I just wanted to go over some of these mutation notes that we won't be covering in tomorrow's web quest. Uh, just go in a little bit more detail than what we've already done. So most of you know a mutation really is just a change in DNA. And there are two basic types. There's somatic mutations. These are the ones that affect your body. They will affect any cell that descends from the body cell that had the mutation, but not transferred to your offspring. For that, we have to have germline mutations that mutate your sperm or eggs. We'll go over a couple of examples of mutations and how they alter protein and what they can do. First one's hemoglobin, carries oxygen to your body. It's made up of 4 proteins. And if there's a mistake, we can go from normal red circular cells to the sickle cell, that these crescent moon things that get caught in arteries and other capillaries and things like that that can cause those vessels to burst. It extends from just one base being changed. So instead of CTC, it's CAC, which translates to a different amino acid. So instead of glutamic acid, that's water-loving, we change it to valine, which hates water. Instead of being these molecules that are floating around in the liquid of the cell, they create these things called sickles that cause a distortion of the red blood cell, all from one gene. So with the homozygotes, they will get the sickle cell anemia disease, which can cause lots of problems there for that person. And generally, when they get in low oxygen conditions, they will definitely have issues. You see more of these sickle cells come about. And this sickling will cause anemia, joint pain, and organ damage. In fact, most people die of kidney damage as these red blood cells get in the vessels and cause those vessels to burst. Thalassemia is another beta globin mutation. In this case, there's too few beta hemoglobin molecules, and that causes too much alpha hemoglobin that says it can't bind to the beta globin and become a hemoglobin molecule. It will release these alpha globins and release that iron, and too much iron will actually be damaging to your organs as well. And if you're just heterozygous, you have what's called thalassemia minor. Those people can survive, but if you got thalassemia major, where it's homozygous, these children die pretty young. Living past age 12 is going to be difficult. Why do we see so many hemoglobin mutations? Basically, it's because of malaria. Malaria is pretty widespread, infects humans, and does basically kills them off. And so there's all these mutations to our hemoglobin to prevent the destruction by the malaria organism. And thalassemia and sickle cell, if you're heterozygous, you're generally immune to malaria. Another one we see a lot of mutations in protein-wise is collagen, which is 60% of your bone, cartilage, skin, tendons, your teeth. They have a very precise structure. Basically, they're in 3 helices combined together. And basically, it's made from a longer precursor that's trimmed off. And if it's not trimmed off, then we've got some issues. So there's a mutation that occurs where the enzyme that trims off these ends has been destroyed. And so instead of making collagen, they got this pro-collagen that doesn't really bind as well as it should. And it creates disorders. And a common disorder we're finding is Ehlers-Danlos Syndrome. So in this case, the skin is very, very stretchy. These people dislocate their joints very easy. I've had a number of students that have had major issues with this disorder. And you can see how stretchy his skin is. You can go on YouTube and look up Ehlers-Danlos things and see all sorts of things people can do. One guy I've seen has taken his neck skin and put it over his nose. Um, look up Rubber Boy and see how he's made a career of being able to dislocate things at will. So there's a lot of disorders, but most people actually it's pretty bad. Um, one of my students had it. She literally goes to children's about once a week, maybe a couple times a week, to just get her ribs popped back in because her ribs are always getting dislocated. Alzheimer's disease also has some mutations associated with it. It's basically in a gene called presenilin 1, and this protein monitors beta amyloid usage. And there's some sort of missense mutation there that basically stops the translation. And this protein's been truncated so it doesn't work correctly. You get too much beta amyloid accumulation, what then leads to the Alzheimer's disorder. So here's an example of the protein presenilin 1. And we find mutations at various spots here. And if they occur at those areas, generally it's been changed to a stop codon. So this part would be missing if stop codon was there. So wherever you see one of these asterisks, there's a mutation there that's truncated that spot. So we do know mutations can cause disease. And you do know that a mutation is a change in DNA. It can occur spontaneously just through replication of our DNA. That's probably the most common form of mutation, is just being spontaneous. But exposure to different chemicals and radiation can cause the same thing. And those chemicals are referred to as mutagens. They create mutations. So a spontaneous mutation is basically an error in DNA replication. See it here where we have an A that matches up to a G. There's this mismatch there that it kind of bubbles up there. Normally, your DNA polymerase would find that, fix it. But if it doesn't, and you go through replication again, you will end up with an altered DNA from that side, because this guy will then match up over here. So at this point, it's permanent. Here, it's not. It could be fixed. Once it's down here, it's a permanent mutation. It's going to be passed on to all those cells. Depending upon the protein, you can have a higher mutation rate. There's all sorts of different coding regions that lend themselves to get a mutation. That must be why hemoglobin has so many and collagen has so many as well. But on average, we see one about every 100,000 times. Not that big a deal. Normally, we can fix most of them. Keep in mind that only 1.5% of our DNA actually codes for a protein. So if it happens in any of that other spots, the other 98.5% doesn't do anything to you. And there are various hotspots, palindromes, where the sequence reads the same way forwards and backwards. Those can occur. Sometimes we see nucleotides repeated over and over. So that'll increase mutation rates. Sometimes they'll be complementarily paired and create what's called a hairpin. And so when the DNA polymerase goes to read this, it'll get to this point and then just skip it. So big pieces can be lopped off there because of that. And if it's a palindrome, or same way forwards and backwards, that tends to cause instability and mutations can occur. Sometimes chromosomes can mispair, and you can see here where it just didn't line up correctly, and it ended up getting more genes when it crossed over. This is probably how we got our color vision. Most mammals only have 2 genes to see for vision. We have 3, so something popped over there so that we ended up with 2 genes. There are things that can induce mutations. They're called carcinogens. So all sorts of things. Probably the most common one's UV radiation. The sun creates thymine dimers. So that's when 2 thymines come together. They're sitting right next to each other on the DNA strand, and they just bind together, creating little bubble issues if it's not fixed. Um, Acridine dyes for a long time. M&Ms didn't sell it. Certain reds and blues because they were acridine dyes and they caused cancer, so they were removed from the market for a long time. X-rays actually break chromosomes and delete nucleotides, so that's why you got to wear all those lead aprons and stuff every time you get an X-ray. Let's see. Ames test, just a way to test the mutagenicity of a substance. I won't go into that now. Exposure to mutagens can cause different things. Probably the most common one is natural sources. This would be things like granite has a lot of uranium and stuff in it, so it can cause cancer. Um, the sun, another big source of it. Medical treatments, probably the next common. When my dad went through prostate cancer, one of the ways they treat it is actually to add radiation to kill off the cancer cells because it kills those faster. So they put these little radioactive seeds in there to prevent the cancer. So it could actually cause cancer. Some of our treatments actually cause cancer. Weapons. Anything that is armor piercing has uranium in it. Most people are thinking we're atomic weapons and things, but really most of our nuclear material comes from these armor piercing weapons. So those tank shells I was using when I was a tank driver in the Army, those things have this uranium, depleted uranium core. So it's not enough uranium to be used in a nuclear power plant, but it's can lead to radiation. So when it's not fired, it's fine. It's coated with something. But once it's fired, radiation is being leaked out. All right. As I mentioned, natural sources is the most common. They're all different types of point mutations. You'll read that tomorrow in the web quest, so I'm not going to go through that. All that'll be there. Pseudogenes are interesting in that they're genes in your body that were once a gene, but they've been removed or they acquire mutations. They don't work anymore. However, a mutation can cause them to get turned on, and often they'll cause problems. And we'll skip all that. And let's see, I do want to point out that not all mutations cause problems. Some of these missense where they're changing one amino acid to another. If you remember your codon code, a lot of them will code for the same amino acid. So it may change it to technically it's a mutation. It may change it so it codes for the same amino acid and therefore it does nothing. Some of the amino acids, if it changes like glutamic acid to aspartic acid, they're very similar chemically. It may not alter function. So you can't really look at the mutation to see if it's going to cause any problems. Some of them are conditioned on things from happening. You won't see the mutation unless this condition comes up. We do have some repair mechanisms. I won't go into much detail on that. DNA polymerase is a big repair mechanism system, but there are other ones. You can look at these notes if you want to learn more. Excision repair, mismatch repair. And if we don't repair them, generally you'll accumulate a protein that will cause you to have your cells go through apoptosis and die off if we don't repair it. However, there's sometimes that this monitoring system doesn't work. These different disorders as well that can create that. Colon cancer. Xeroderma Pigmentosa. Here's an example of that. So these are basically, this is a UV radiation causing thymine dimers. The protein that's supposed to cut those out doesn't work right. And so when you go through replication, hit one of those thymine dimers, you stop replicating your DNA. So that cell can't function anymore. And so it doesn't grow. So you'll get these little scabs and things because your cells just aren't growing right. Goes in. And you can see that this is UV radiation. Right, you can see where his t-shirt is worn. He doesn't have as much scabbing here. under his neck where he's not going to receive the UV radiation either is fine.